Welcome, everyone. Good to see a lot of familiar faces, and especially Michael, who we're saying has the distinction of being, after 2 years of the series, Michael's our first repeat offender. Um, and we're very happy. He shared with us that the data he's going to show us today is all from his new lab out at UCLA. And thanks for getting in touch to give another seminar, and very happy to have you. Thanks so much, Michael. Take it away.
Yeah, thanks so much for letting me uh, give another talk here. This will be a little bit different from the first time I spoke. The first time I spoke, it was focusing on villages as a whole, sort of our plans for villages and a little bit of how we make them. Today, it's going to be a little bit more focused on a specific story that we just posted to bioRxiv about a month ago. And because it's more focused, it'll also be shorter. So this is only about 20, 21 slides. And so hopefully we'll have ample opportunity for conversation. So
Great.
today we're going to talk about how we have adapted the village system to perform self-fitness screens and how we then use the output from these screens to perform GWAS and ADISH analyses to identify both rare and common variants that are associated with self-fitness. So let's get started here. So I like to say that every single one of us in this virtual room are here because a small clump of cells were able to proliferate and persist. Now, I'm of course referring to how immediately after conception and lasting throughout our development and maturation, the cells in our body must be able to increase in number over time in a controlled manner and also remain viable in the face of many internal and external stressors. Now, collectively, for the purpose of this talk, we'll refer to these processes as cell fitness. So my lab is particularly interested in the fitness of fetal neuroprogenitor cells, which of course are the cells that proliferate and differentiate to give rise to the many different neuronal and glial cell types that populate the postnatal cortex. And because of this early and critical role in brain development, these cells have a major influence in how we think and how we behave and socialize. And they also play an incredibly important role in the onset of certain conditions like autism. We know that both genetic and environmental factors can influence NPCs and their traits and their activities. So we're aware of common and rare variants that can modulate proliferation and fitness characteristics of these cells. And we also know we're actually keenly aware of how certain environmental stressors like viruses and toxicants can disrupt neuroprogenitor cells and alter the developmental trajectories for individuals. Now, understanding how these genetic and environmental factors, as well as how they interact, how these actually impact neuroprogenitor cells and influence the developmental trajectories, this has been a bit of a challenge for a couple of reasons. So first, fetal brain cells are largely inaccessible, and I'll talk a bit about how we have gotten around that inaccessibility. It's difficult to obviously study human brain cells from living people or from even postmortem tissue. Also, something that's really important to keep in mind is that the conventional G by E methods, so gene bioenvironment methods that do rely on human subjects, there are certain limitations to these approaches. So they tend to require thousands of samples. They lack temporal or cellular resolution, so they might identify genetic loci associated with certain traits, but we don't necessarily know offhand what cell types are impacted, nor do we know at what stage of development those variants may be exerting their effects. Furthermore, one of the most difficult things with G by E studies from human subjects is how difficult it is to actually quantify environmental factors. Often we have to use proxy measures like their address, and we can say like, okay, based on where they live at this given time, we're estimating the level of exposure to say air pollution or to lead. It can be quite difficult to quantify that as well as to deal with the fact that we're not exposed to one thing and we're not exposed to one thing at a time. The exposome is quite complex. And so trying to disentangle those environmental contributions can be quite difficult from these human subject studies. And also, we tend to get minimal insight into mechanisms from the GWAS alone. And so, in fact, we know of about only 20% of the GWAS loci are even known to be EQTLs. And so it's a lot of missing heritability when it comes to these GWAS studies that still need to be interrogated. So how do we try to overcome some of these limitations? Well, one strategy, as I don't need to explain to this group, is the use of human stem cell-based models of the prenatal brain. As we're all aware, we can take these stem cells, either from embryonic stem cells or from induced pluripotent stem cells, and convert them to many different cell types that populate the brain. We can even generate these cerebral organoids or cortical organoids to study multiple cell types at a time that are building structures that are reminiscent of what we see in vivo. So my lab actually focuses on neuroprogenitor cells. I'm not going to go too much into the protocol because I did mention it in my last talk, but I'm more than happy to explain a little bit more in detail. Essentially, we introduce a doxycycline-inducible version of neurogenin 2. We then turn that on for anywhere from 24 to 48 hours. It's a quick pulse of NGN2 overexpression. And then we can move the cells over to a neuroprogenitor cell maintenance media. And through that process, we can make NPCs from stem cells in a highly robust and very quick way. To date, we have generated 217 lines. So 217 stem cell lines that we've converted to neuroprogenitor cells using our protocol. And again, I can explain that more in detail. We've published both a paper on it. It's part of one of our papers, and it's also a protocol paper that I can share with people if they're interested. So with this in mind, we should also talk about the limitations of the human in vitro studies. They're not perfect, and they're in my opinion, able to overcome some of the limitations of human subject GWASs, but of course, there's still many things we should consider here. So first of all, the typical approach to studying these human in vitro, to perform these human in vitro studies is to involve a small number of genetically distinct cell lines. So these studies historically have been underpowered when it comes to understanding how genetic variation can influence some of the neuroprogenitor cell traits you may be interested in. And the other issue is that when we are trying to study many cell lines simultaneously, your incubator might look something like this, where you have each cell line cultured separately. We call this the arrayed format. This can introduce quite a bit of well-to-well and plate-to-plate variation that can actually mask biological signal, especially if it's a subtle yet still important difference between your groups or your cell lines. Also, this can be quite low throughput in the sense that it's costly, it's time consuming, and it's laborious if you're trying to do all this and keep all these cell lines alive for a long time simultaneously. Really, the issue here is if you're trying to do a G by E interaction study in which you have multiple donors and you have multiple perturbations or exposures, different concentrations, you can see how the number of samples you need to process can get out of hand fairly quickly if you want to be well powered. And so, as I've talked about in the past, our solution to these three issues here is to just throw them all into the same dish. And that's what we call our cell village. And so what cell villages essentially enable are these population genetic studies in a dish. So we can start off with dozens to hundreds of different donors. For each of these donors, we genotype them ahead of time because we actually need that genotypic information for later on when we're trying to re-identify each donor from our molecular and cellular assays. Now, what I also want to emphasize here, and again, we can talk about this more if you're interested, is what we do in my lab is we actually generate each of these cell lines separately. So when we do the induction to NPCs or to neurons or to glia, we actually do them in an arrayed format. And then we pool them once we've already done QC on each of the lines independently. We just think that's really important because if not, you can get some pretty gnarly donor imbalance. If you're trying to do the inductions while they're in a pool together, they can grow at different rates, and that can really throw off what you get at the end. We also have to contend with cell type contamination. If you're trying to study neuroprogenitors, for example, but you happen to have a line that you threw in there that doesn't make NPCs very well, or let's say it's more prone to becoming a postmitotic neuron or even going down like a neural crest lineage, that means your village won't be a pure cell type population, and that can influence some of your sensitivity for your assays. So the real key here is once we have all of these and we've captured this genetic and molecular and cellular phenotypic variation in this shared environment, We essentially minimize the technical noise, and then we can perform these types of experiments here. Because they're in a uniform environment, now we can challenge them with different perturbations and ask questions about how natural genetic variation across our donors actually influences their response to these different stimuli. So that stimuli can be something endogenous, like signaling molecules, sonic hedgehog or Wnt, or it could be external stressors like viruses and toxicants. We can even use this platform to ask questions about drug safety and drug efficacy using this village system. Okay, so we're going to focus today on cell fitness. I refer to this as both collectively as proliferation and viability in the face of stressors. And so, regardless of which assay we're actually performing here, I want to get a few things out of the way so you can understand how exactly we're doing this. So, if you're wondering, well, how do we know each donor's fitness or proliferation rate if they're all mixed together? What we're going to do here is use a tool called SensaSeq. Now, what SensaSeq does is it looks at the donors in the village. So we just harvest bulk DNA from the village. We perform low coverage whole genome sequencing, like 0.5x, 1x coverage, costs about $100 a sample, depending on who's doing the sequencing for you. And then what SensaSeq does is it tells us what are the proportions of each donor in that village. So, for example, if you have 100 donors and they're all in there equally, well, then each donor should be 1% of the village, and SensaSeq should tell you that each donor is 1% of the village. Now, what we can do is we can harvest village cells at different times, so across time, and ask questions about how those donor proportions change. So if donor one is a faster grower than donor two, well, its proportion should increase over time. Whereas, let's say donor two is a slow grower, its proportion should decrease over time. What we can do is we can actually convert those to proliferation rates based on that information. So a few really important things I want to highlight here when trying to establish this village-based assay had to do with some common questions we actually have received over many years when describing cell villages. And so I want to show you some of these validation experiments. Again, all of these are in the preprint except for the last panel here, and I'll describe that in a little bit more detail since you can't find it anywhere else at the moment. So the first thing we wanted to address was this question about whether or not villages capture cell intrinsic traits. In other words, are you measuring something that's intrinsic to that donor, or are you simply measuring a bunch of cell non-autonomous effects that just come about from donor A influencing donor B and so forth? So to address this specifically with proliferation, what we did was we took 12 donors and we plated those 12 donors in an arrayed format, and we used traditional imaging-based metrics to count cells over time and calculate growth rates for each donor. We also simultaneously, we took those same 12 donors and pooled them in a village, and then used our SensaSeq readouts to measure each donor's change in proportion and then convert that to proliferation rates. And so you can see here, we actually compared those two. Our R value is approaching 0.9. So we have a really strong correlation between our village-based metrics and our arrayed-based metrics, suggesting that for the most part, we are indeed capturing cell intrinsic proliferation in this village system. Next, I want to highlight that the well-to-well variation is indeed decreased in the village system compared to the array. I want to highlight as well that our batch-to-batch reproducibility is quite high. So in this experiment, we made a village of 12 donors and did SensaSeq readouts over time. We made the same exact village, I believe 5 days later. So we repooled them and did everything again and measured growth over time for each donor. And as you can see, again, very high R value, suggesting high correlation between batch one and batch two in this setup. And finally, this is something we haven't put in the preprint. We're working on something else right now to include this data. This is, I think, very important for people who might be scared away by the idea of, wait, you're telling me every single time I want to do an experiment, I have to culture these cells simultaneously for a few days before pooling them. What we're finding is that these villages, at least the neuroprogenitor cell villages, do quite well with freeze thaw. And so in this experiment, I think this is a 35 donor village. What we did here is we plated that village, and did census seek readout, in real time. So the cells were plated live, and we did census seek over time. And what we also did is at the time that we pooled those donors, instead of plating, we froze them down. And then 5 days later, we thawed them and again did census seek over time. And as you can see, the donor proportions either being plated immediately or being plated after a freeze thaw, again, they're highly correlated, suggesting that our donors are able to equally survive the freeze thaw and maintain their relative growth rates to each other. So this is really nice because it suggests that we don't have to make a village every single time. We can actually bank down, dozens of vials whenever we make a village. It also suggests that we can make larger villages by sort of pooling and multiple mini pools, meaning let's say you have three mini pools of 50 donors each that you froze over multiple different times. Instead of waking up 150 lines, you wake up those three vials, pool them, and then plate them down. So now you have all 150 represented in a single village. So we're actively working on these sort of mini pool approach, so we can sort of mix and match over time depending on which experiments we want to run and build larger villages in that way. So this is where it gets a little complicated. Um, it was around this time when we were working with our collaborator Harold Pimentel here at UCLA, he's this expert bioinformatician and methods developer. He pointed out something very important about census-seq data as itself. He mentioned that census-seq data is compositional, meaning that all the values must add up to one. The problem with that is it violates the independence assumption of most linear models. Now, what I mean by this is even in the absence of any sort of non-autonomous biological influence, the way we're actually reading out donor proportions, they are influencing each other. So if you have this example at time 0, you have 3 donors, they're all equal to each other. Let's say donor 1 is faster than donors 2 and 3. Well, it will increase its proportion, in this case up to 60%, while the others will decrease their proportion. Again, even if donor 1 is not actually changing the growth rates of donors 2 and 3. The way we're reading it out means that they're not truly independent of each other. Now, this is fine. It just means that we have to use specialized tools that are adapted and created specifically for this sort of compositional data. And so with that in mind, Harold and his grad student, Chloe Hansen, developed this tool that they call Townlet, which is a Dirichlet-based model for census-seq compositional data analysis. So the inputs include taking your census-seq readouts, which are these sort of each donor's proportion across your time points or across your treatments. You also let the system know, the different time points, different treatments, how many replicates you have. And then from there, it can give you both donor specific effects as well as group effects. It could even identify certain sources of variation. And so essentially what this tool is doing is it's properly accounting for this compositional nature of the data, and so that the analysis we're performing is not violating any major rules of the tools that we're trying to use. There's a lot of simulations and deep characterization of Townlet in the preprint. I'm just going to give some quick highlights in this slide here, and again, we can talk more later if you're interested. What I really want to highlight here is that in the panel on the left, you can see that Townlet has higher sensitivity relative to other benchmark models. So we have the frequentist Dirichlet, which is in that teal color. The green is exponential growth rate equations, and the purple is the linear model equations. These are all from simulations, and what I also want to highlight here is what we have is D is the number of donors, R is the number of replicates, and T is the number of time points. What we want to highlight here is that with more donors, you actually increase sensitivity. And we also want to highlight that regardless of the number of donors, Townlet outperforms all these other metrics or all these other tools when trying to quantify, when trying to detect sensitivity. I also want to point out on the right side that Townlet also has a lower false discovery rate compared to ANOVA-based approaches. And so on these two metrics, these two very important metrics, Townlet is outperforming other tools. And so we have a lot of confidence moving forward with this tool given the results of these early simulations. Okay, now I'm going to give you 3 use cases that I hope will be the launching point for many other um, future projects, both in my lab and others, that leverages this village plus townlet platform. So the first thing I'm going to talk about is 16p11.2 deletion. And this is an example of how a rare copy number variant can influence and modify neuroprogenitor cell proliferation. So one thing to highlight about 16p is that it's a major risk factor for autism, intellectual disability, as well as enlarged cortical surface area and macrocephaly in general. Now, some of the earliest studies were suggesting that, or at least hypothesizing that increased neuroprogenitor cell proliferation was the driving force underlying this brain overgrowth that was observed in anywhere from 30 to 40% of the patient population. However, the early studies have also been quite contradictory. Some mouse studies found that there appeared to be increased proliferation of this cell type. There was one iPSC-derived study that also showed this, but others showing that there were no differences in growth rates between patients and neurotypical controls. So one possibility for this sort of these contradictory studies is that they tend to have small sample sizes. So, a small number of donors, we're talking 3 to 4 typically. And we also need to be keenly aware of the fact that the patient population is highly heterogeneous. There's a lot of variation across these individuals in terms of their symptom severity. And again, I mentioned that only 30 to 40% even present with macrocephaly. So it can be very difficult to model if we're only using a small number of donors. Also, keep in mind that this is not cancer necessarily, so we're not expecting a doubling or tripling of growth rates. The effect itself might be quite subtle, and this could be lost in certain arrayed platforms if there is high technical noise that's actually masking the subtle signal. So what we did is we took every single patient line we could get our hands on at the time. These are all from the Simons Foundation. I think there's 2 or 3 more that are now available that we're in the process of acquiring. And we compared these to a similar number of controls. These are all neuroprogenitors. And so what we did is we harvested over multiple time points, multiple replicates per time point, and we plugged them into the townlet model. And so what we're seeing here is that a positive value indicates a higher proliferation rate. Negative value means lower proliferation rate relative to a baseline donor. So you can see each donor here color coded. And what we see on the far right is the actual the actual analysis, the statistical analysis that Townlet provides, in which it's showing us that the effect of the deletion is positive and significant, suggesting that the CNV actually does lead to increased proliferation of that population relative to the neurotypical controls. I also want to highlight, I don't show this here, but it is in the preprint. If we get rid of that NFID on the far right, that donor on the far right, even if we get rid of that donor, we're still seeing the significant difference. We also wanted to validate this study, and so we validated the sort of village approach by showing that if we cultured all of these donors separately in an arrayed format, and again used traditional imaging-based readouts of growth, essentially we just count the number of cells over time and see, and then calculate growth rates from that. Again, we're seeing this significant difference in which the patients are hyperproliferative. We even got our hands on an isogenic control from Mike Tolkowski's lab. And again, we're seeing that the deletion leads to a hyperproliferation of these cells.
Hey,
So,
Michael, I
yes,
got a quick question.
yes.
Both on the data you showed on the previous slide and then the arrayed validation, did you find that, and you mentioned this specifically, that 30 to 40% of the 16p11.2 deletion patients have macrocephaly? Do their
Mhm.
in vitro growth rates correlate with that phenotype?
We're trying to track that down.
Okay.
Um, so I should mention, let me show you if I can go back. Safari does a really good job of providing some of that information about the actual phenotypes, symptoms in the patients. Uh, One thing I should also mention, there's actually the NFID line,
Hmm.
and I think one of the other lines I can't remember. The NFID we actually have no information from. That actually didn't come from Safari. That came from the Broad Institute, and they have next to nothing on that person about
Cool.
their symptom profile. I think one of these lines we actually reprogrammed ourselves, and that is also a line we don't have all the information for. So the issues are a little underpowered in terms of knowing
Yep.
all the details about these individuals. And again, Safari, I think they do a good job of this, but if you look through some of their profiling, there's a lot of gaps. And so it really depends on what that specific doctor for that patient was actually measured.
Recording.
Yeah.
Gotcha.
Even for so most of the controls, not all, are coming from the CERM biobank, the now defunct CERM biobank. Um, Even there, they do provide head circumference for a fraction of the donors. And so we are trying to see, like, of the ones we have that information on, do we see any relationships? But again, and it's just a little tough because, one of the things that we know about this sort of macrocephaly, at least commonly in autism, is that the macrocephaly tends to present in the first year or two of life. It doesn't necessarily maintain once they reach adulthood. And so, because a
Hmm.
lot of these serum donors are already adults, their head circumference doesn't tell us that much about whether or not they presented with macrocephaly in the first year or two of life. So, it's a little tricky. I do think there's been other work. I think Eric Krechesny has a paper out with a bunch of people. I believe it was maybe it's a PN, a molecular psychiatry paper, in which they did collect iPSCs from individuals with autism who do present with macrocephaly, and they showed that they had a higher NPC growth rate relative to neurotypical controls who did not present with any sort of increased head size. So we're not necessarily powered to show that, but in that study, that's exactly what they're trying to read out. And so they did have very detailed information about those individuals in their macrocephaly.
We also show that in idiopathic individual with microcephaly,
Yes.
even without the deletion,
Yes,
that
exactly.
there is an NPC are slightly have a slightly higher proliferation. But I was going to say that the question of whether the macrocephaly is maintained or not in adulthood is controversial.
Mm,
And the problem is there that most of the studies are cross-sectional and they're not longitudinal. So
yes.
when you have cross-sectional studies, it's really difficult because of this inter-individual variation that you have. So I'm actually not sure whether a true, well-powered longitudinal study has been done
Yeah.
in macrocephaly in
Hmm.
general. Yeah.
Right.
Because
Yeah.
not only all macrocephalic, of course, have this deletion. So yeah.
Exactly. Yeah. And I'm kind of experiencing that. I have a 3-year-old son, and when we were in parenting class, and their parents coming and saying, like, they knew I studied autism, like, hey, my son is presenting with
Oh,
a head
yeah.
circumference in 99 percentile. Should I be worried? And, like, we just don't know enough to say that that's something you should immediately be worried
Yeah.
about.
Totally right.
No,
Yeah.
no, no, no. Have you read the papers of Bob Schultz?
Mm,
Macrocephaly in the normal population is an adaptive trait. My daughter also is at the 97 percentile.
mhm. Yeah. Yeah.
It's a complicated issue.
Yeah. Yeah.
Exactly. Yeah. Cool. So did I address that,
Oh,
Carla?
yeah. 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 Cool.
Yeah. Where am I? Let's see. Okay. So yeah, just to kind of close this out, we're really excited about this. Just the ability to measure this. Of course, proliferation is a relatively simple phenotype, and I'd love to talk more about other things we're hoping to other types of cellular phenotypes we're interested in studying. But I think this is a good start for starting to investigate either known patients with known mutations associated with autism, or even idiopathic versus control experiments in which we are looking for differences in proliferation. I think this really highlights the ability to do group analysis, group-based analysis with the village, which obviously is going to be better, you're typically going to be better powered than these sort of common variant studies, which I will show you now. So we then wanted to ask a question about whether or not we could identify common variants that modify or that is associated with NPC proliferation. And so I'll be quite honest with you. When we first did this, this was meant to be a pilot study to see what sample size we would need in order to detect some of these common variants. And it turns out this pilot quickly turned into the actual experiment because we were well powered with only 35 donors. So let me explain this a little bit to you. These are 35 embryonic stem cell derived NPCs. We harvested at day 0. So this is sort of our, before we plate the cells, they're all pooled in a falcon tube. Before we plate them, we take a sample just to make sure we put them all in the village at equal proportions, or at least if we didn't, we need to know what those starting proportions are. We then harvested after 1, 5, 6, and 10 days. For each of these time points, we harvested 3 samples because Townlet really performs well when you have multiple replicates, which is something that we were sort of testing out in the simulations. And 3 seems to be enough to get good results out of this. So what we do here is we converted those into a growth rate. And so what I want to highlight really quickly is you can see for each donor, they're actually presented as a distribution. What I like about this system is we can collapse all these data points into a single distribution and then use the mean of that distribution as our phenotypic score. This is important to us because initially we were concerned, like, okay, do we run a GWAS on day 5 versus day 0, or day 6 versus day 1, or day 10 versus day 5? It was just, you could p-hack your way to a result if you just decide which of these comparisons actually yields the most variable effect scores across your donors. So we're trying to be very sensitive to that and make sure that we are just capturing all the information into a single value for each donor. So you can see quite variable effects across our donors. Negative value, again, corresponds to slow growth compared to positive values. And then we just ran a GWAS on this. This is the phenotypic score. We already had all the genotypic data for each donor going into the experiment. You can see a couple of those blue dots are corresponding to variants and loci. So loci that reach suggestive significance. But then we have that locus on the 16th chromosome that maps to the ZFHX3 gene. And this actually did reach genome-wide significance. So let me talk a little bit about that gene. So first, what was very interesting to us is this gene tends to be preferentially expressed. It's actually enriched in the earliest time points of brain development. It actually sort of goes away once you get to the 3rd trimester. 2 of the other genes that were suggested that were interesting to us, MSI2 and MYT1L, those tend to have expression throughout life in the brain, whereas this ZFHX3 was enriched. The actual effect was quite interesting to us. So this is the index SNP that was identified through this GWAS. You can see donors that have the 2 copies of the reference allele, the TT genotype. They have a higher proliferation effect score compared to donors that have the T with an insertion. So the other allele has an extra T present. I want to highlight that other work has shown that in mouse models, actually, that this gene does play a role in differentiation, so neurodifferentiation. And so it essentially acts to help the cell exit the cell cycle. And so there's already precedent for this gene playing a role in some of these neurogenesis type phenotypes. And so what we're saying here is that this appears to be the first study to suggest that natural variation in the genetics of this gene appear to be playing a role as well in natural variation in NPC proliferation across our donors. I also want to highlight that that lead SNP there, you can see in the purple. You can see this cursor? I guess my cursor's not visible. With the giant red arrow at the bottom, pointing to that purple diamond. That is our lead SNP. As you can see, it's actually located in an enhancer region for ZFHX3, suggesting that there is some sort of functional impact or some functional role of this variant in this gene. So we're pretty excited about this. We're in the process of starting to validate some of these results. Essentially, we're going to toggle ZFHX3 expression in some of our slow growers and some of our fast growers and see if modulating expression of this gene does indeed change the proliferation rates of these different donors that appear to be showing differential growth rates that associates with this genotype. Any questions on this before I move on to the next and final use case? I see. Okay, cool. We can come back to this if there's any other questions. Last story has to do with genetic modifiers of environmental stressors, so in response to environmental stressors. In this case, we chose lead sensitivity. We're very interested in lead. We actually started this work about maybe 2.5 years ago. I gave a talk here at UCLA, and Patrick Allard, who's a expert developmental toxicologist, came up to me afterwards after he saw my story about Zika virus infectivity and said, "Hey, you could apply this exact same framework to studying toxicants and their impact on the developing brain." And so we're essentially asking questions here about, is there differential response or differential sensitivity to lead across our donors? And if there is differential sensitivity, can we find any genetic biomarkers of that sensitivity? And so again, we have that same exact 35 donor village. We hit it with two different concentrations of lead, which I should mention we've known for 2,000 or 3,000 years now that lead has adverse effects on the brain, both adult and developing. And so we have these two different concentrations harvested at two different time points. And again, we're able to collapse all that data into a single distribution for each donor. And then we use the mean of that distribution as our phenotypic score. In this case, negative lead effect value indicates the donor is more sensitive. A positive value indicates the donor is more resistant to lead. And so with this 35 donor village, we did not reach genome-wide significance for any locus, but you can see with that ARNT2 locus is just slightly below that threshold. And so we wanted to at least look into this a little bit more to see if there's any biological potential there for discovery. And so one thing that we first discovered about ARNT2 is that it's a transcription factor that's actually known to be neuroprotective. It's actually enriched in the central nervous system. It's very lowly expressed in other parts of the body. And what has been shown is that it's believed to protect cells from hypoxic conditions. And it's quite understudied. And the idea is that perhaps it plays a role in protecting cells from other sorts of environmental stimuli. The other thing to keep in mind is that lead has recently been shown to induce pseudohypoxic conditions in cells. So it is possible ARNT2 is playing a protective role through those mechanisms. Now, this lead SNP here, it's not as clean cut as a story as I just described to you with the proliferation. In this case, it is mapping to the protein coding version of this gene. There's actually two SNPs that were in high LD with each other and perfect LD with each other that had the highest that was the highest in that Manhattan plot. And one is in an enhancer region of the protein coding gene, and another is actually in a divergent transcript version of this gene. Divergent transcripts are believed to play regulatory roles in the amount of expression of the protein coding gene. I'll say that we have found that I think it was in GTEx, showed that this variant that's highlighted with that red arrow is an EQTL in adult brain tissue. We're still parsing through single cell data that we generated from this to determine whether or not this is indeed an EQTL in our data set. But our preliminary analysis suggests that it's not. And so what's interesting to us is that perhaps this is actually a response EQTL. So more than happy to talk about response EQTLs because something we're getting increasingly interested in. Only about 20% of GWAS, human subject GWAS hits are actually identified as EQTLs. It's believed to be that many others could potentially be response QTLs, and so perhaps this is an example of one of those that's showing up where on the surface we can't exactly explain its functional role, but with increased scrutiny of response elements, maybe we can identify something there. The key is, though, at the very least, that variant that's denoted with the upside down red triangle does appear to be in a regulatory region of the protein coding gene. And so we do have some confidence that at least the protein coding gene is being regulated in some way. And just on the far right, I want to highlight the effect is quite strong here. So genotype at this site explains about 55% of the variation in our lead effect. This is approaching almost identical effect size as the IFID-M3 variant we previously described in our cell stem cell paper that talked about risk variants for Zika virus infectivity, which we also were able to detect from a relatively small village. I think there's only 41 donors in that village. And so this really is highlighting how the village system is able to identify variants that are potentially associated with response to different environmental triggers. So with that, we have about 15 minutes left. Just want to acknowledge people in my lab, our funders at CIRM, NIMH, and Simons Foundation, as well as internal funding from the Broad Stem Cell Center. I want to highlight Harold and Chloe, who built the Talent model with us, and then Patrick, who's been instrumental to our foray into environmental toxicology. And then I will leave this up here. Maybe I'll actually link to this paper in the chat. That's probably easier. So if people are interested, they can just click on that rather than trying to take a picture of their screen.
Great.
Link to that way.
Yeah, we have a link on the
Right.
seminar site too, but that's great. Yeah, make as
Cool.
easy as good. Thank
Cool.
you.
Yes, of course.
Questions? I think there'll probably be many. I have many.
I have a question.
Sure.
Can I ask a question? This is so powerful. It's really exciting. And of course, we're used to GWAS with thousands of participants, and so why is this so powerful that you can see things with fewer donors?
Yeah. This is my favorite question to answer because I think it gets everybody excited when we talk about it. Um, I think it's because for our cultures, we really, really aim for cell type purity. So it's a single cell type. It's a single phenotype we're trying to read out, whether it be growth or, death in the case of lead. And there's essentially no technical noise because you've gotten rid of the well to well, batch to batch, plate to plate. And also when you create when you're harvesting for library preparations, whether it be DNA library prep or RNA library prep, again, that's all being done simultaneously. All the donors are being treated the exact same way throughout the entire pipeline. And so I think that's primarily why we're able to detect these effects. Think about human subjects. That's a complex person that you're looking at different points in their life, different exposures, different everything. And even the way we're trying to measure some of these traits is not necessarily precise or accurate. I mean, I think even, when it comes to, say, autism or schizophrenia, the way people are diagnosed could be very different depending on who their doctors are or where they live, their access to healthcare. So I think the messiness of those human subject approaches, just the natural inherent noise to those systems, is why they require so many subjects. And our elimination of that noise, I think, makes it much easier for us to detect these things with, pretty small sample sizes.
Beautiful. Thank you.
I think is he Shiraz with the hand up?
Yes. Yeah.
Beautiful talk. And it kind of reminded me, right, at its premise of using like deconvolution methods for bulk RNA-seq when you're
Yes.
trying to identify different cell type proportions. And similar to the flaw that you pointed out early on, there's also a violation of the law of independence across different cell types, right? But there are a lot of methods in deconvolution that embrace that violation and go the route of nonlinearity. So it's not so much a question, but more of like a comment on
Mhm.
if you could maybe talk a little bit more about how you arrived at this hierarchical model in Townlet. Did you test different methods and what made you decide on moving forward with this particular non-parametric assuming model?
That's a great question for Harold, I will say. Yeah, so my understanding is very early on, they were doing a lot of whiteboarding essentially. I don't even know if they were necessarily testing out different models beyond the ones that were shown in that figure. Um, and so I don't actually know a lot about the history of where they came up with that. I know almost immediately that when they saw this data, they're like, oh, this needs to be a Dirichlet model. That was their first indicate that first sort of reaction. And my sort of reaction was, I don't know what that word means. Please explain. And so, yeah, it's a good question. I can talk to them about the sort of logic behind that one. It could have been one of those things where they tried it and it worked pretty well, so they just stuck with it. Yeah. That's a great question that I probably shouldn't have the answer to at this
Yeah.
stage.
No, I mean, it was a great talk, though. Thank you so much.
Yeah. I see Sarah with her hand up.
Yeah.
Great talk. Really naive question. Can you elaborate more what you mean about response QTL?
Yes, sorry about that. I wonder if I have a slide. You know what? I will just use my hands. How about that? So there's many ways of describing this. I'll give like the simplest description of a response QTL. Imagine there's a genetic variant in which your genotype at that site has a relationship with the expression of a nearby gene. So that's just like basic QTL, right? So let's say, sorry, let me wonder if I could share this again. I'm a very visual person, so maybe again, I'll just try. I don't have a good example of this. Let's say, and I can't even see myself. So let me see if I can see myself. Where's self view? Show self view. Okay, so let's say this is the relationship. Here are 3 different genotypes on this x-axis, and the y-axis is expression of a nearby gene. If this line is flat, it means there's no relationship between your genotype at that site and expression of that gene, okay? If it's like this, right, now your genotype actually has an association with expression of that gene. That's what we call an expression quantitative trait loci. So a response QTL will be a situation in which at baseline, there's no relationship, but you hit it with lead and all of a sudden there's a relationship. And so the idea is that perhaps a lot of the missing heritability that we're trying to extract from GWAS studies are actually these response QTLs that unless you stimulate the cell or the tissue with this specific exposure, you're actually not going to see the relationship between genotype and expression at that nearby gene. And there's been some studies showing that, hey, when we did this looking at baseline, we could explain 0.4% of the GWAS hits were because of UQTLs, but then when we did stimulating with various things, now 4% of the GWAS hits could be identified or could be explained as response QTL. So there's more and more studies saying or suggesting that we can actually find much more if we look at specific perturbations and ask about how genotype influences response to those perturbations.
And do you think this missing heritability could be due, for example, to epigenetic modification specifically in that loci?
Perhaps, yeah. That's one possibility. I know we can also do what we call CA-QTLs, so chromatin accessibility QTLs, and then response CA-QTLs, in which you're looking at how the chromatin accessibility may change in response to genotype and perturbation. And so I wouldn't be surprised if we identify even more of the missing heritability as we continue to look at these multiple different layers of response. We're just now
Can you
getting
show that actually?
Say
I
that-
mean, because it's an epigenetic response, really, isn't it? And does it have to be stable,
Does it
right?
have?
Do you see what it's a very interesting idea. I mean, you see it in other examples in psychiatric disorders, where particularly, I think, in response to stress, cortisol levels, right, mark an epigenetic response in people who show PTSD. I think it's a really interesting topic you're discussing.
Yeah, I think Kristen Brennan's been doing a lot of work on that. I don't know if it's published yet, but it's definitely pre-printed where she's showing how the response appears to be different and that there's actually genetic markers of who's going to show that variable response.
You know, Kristen's work is beautiful.
Yeah.
But there's also work from European groups along the same lines where they've actually measured cortisol levels longitudinally.
Yes.
And you can see it's not the base level, you show elevated cortisol levels in the serum in response to a stressor long term.
Yes.
So to go back to your question, Sarah, I think the reason I'm stressing it is because it's actually essentially pointing out a wider epigenetic switch rather than a simple. We tend to think of GWAS as being a snip in a particular location, whereas this actually could be a really powerful and large. It could go back to the question of why do you see these changes as well, but I don't quite understand yet how you can see such stable effects in such small numbers
Mm.
of
Could
cell
also,
lines.
sorry, no, it's
Yeah,
okay.
go for it. Yeah.
I was wondering whether this could also fit into the general discussion of what's an enhancer, and are all enhancer just basically active or non-active, or is it only active in the presence of a stimulus? You know, as a more general way of understanding enhancer activations. So you need not just the intrinsic milieu, but something temporally modifying, inducing some sort of signal transduction pathway or something that actually triggers that activation. I mean, it seems to me something that worth exploring, but probably not easy.
Yeah, it's definitely something we're getting more interested in. There's a few technical things. We realized like, hey, we really should start looking at chromatin accessibility, given the multi-ohm kits are becoming more and more accessible. But it wasn't until the BD Rhapsody system sort of built-in single set ATAC-seq that allowed us to financially be able to do this at scale. I think their kit is like 1,800 bucks a sample, and we're getting about 50 to 75,000 cells per sample. So this is going to allow us to really start to analyze villages at both of these levels that we were missing in our first, 2 or 3 years of running these experiments. So hopefully early next year, we'll be able to start reading out some of these chromatin changes.
Could I ask another question,
Go for it.
Carla? Would you mind?
No, go for it. Yeah.
What I wanted to ask you, it's along the lines of neurogenin. So neurogenin 2 is expressed in dorsal uh, telencephalic phase,
Yeah.
and much lower levels in ventral phase. So what I was wondering, I mean, to put it in a sort of like relatively neutral way, is, I mean, have you characterized the type of neuron that's coming out of these systems? Not so much that I care about that, but just to try and understand what the cell number result is telling us.
Yeah, so one thing I didn't highlight is that we actually pattern these at the same time that we're hitting them with the docks to turn on the NGN2. So we are intentionally making dorsal telencephalic NPCs. And I can link to the paper, our cell stem cell paper, that does a pretty thorough characterization of the cell types we're generating there. And we think this is super important because without the patterning factors, people found that NGN2 alone can generate a heterogeneous neuron cell types. You can actually add patterning factors to make, motor neurons intentionally with NGN2. There's a paper out there saying you can add NGN2 with certain patterning factors to make dopaminergic neurons. And so we are really trying to make in this one specific cell type. I will highlight something that's not in our paper, the paper from 2023. We have a newer way of making our NPCs. And when I mentioned we've done this on 200 and something lines, it's actually with this newer method. And that's where we insert the NGN2 cassette into the AAVS1 safe harbor locus. And then do a lot of selection. So we're actually getting even more pure cells than anything I showed you here or anything we've published. And that also allows us, if in the future we want to make, say, DA neurons or motor neurons, we take the same exact stem cells that have the NGN2 already in them, and we simply change the patterning factors that we hit them with during the early induction phase. So we will have a lot of versatility with these cells moving forward. But in the meantime, we're making dorsal telencephalic NPCs. We do single cell readouts, compare them to in vivo cell types to make sure that we are making the cell type we think we're making. And that's pretty much how we've been handling that.
So what factor are you hitting them with to do that?
Yes, XAV, LDN, and SB4 or something. I forget all the digits after it, but it's the same cocktail. One of the same cocktails people have been using for making dorsal telencephalic postmitotic neurons. So we're all in MA's protocol for making those more enriched excitatory neuron cell types. Our initial NPC induction was simply taking that exact protocol and just truncating it down to 48 hours of dox treatment instead of the 7 days. And that's how we're able to
So you get
harvest.
precursors instead of
Yeah,
postmitotic.
exactly. Yeah.
Does it actually reflect, because the protocol is so, how can I say, you're targeting for uniform response, but does it actually reflect inter-individual variation? Have you looked at that? I mean, or not? Because, yeah, I mean,
You mean in terms of like them all
well, I
hitting
mean,
the
I'm
same
worried a
point?
little bit about your readout that
Yeah.
is one cell type made in a very specific way.
Mhm.
And, whereas we're not that, right?
Right. Yeah, so we will miss if there's any, and there is obviously inter-individual variation in their ability to go from stem cells to NPCs. We're missing all of that, right? Like we're just normalizing that to get to the same cell type. So anything that happens before that, yeah, we're not going to be able to actually capture that variation, which is obviously, something very important to keep in mind because, like you said, in the case of humans running around, we don't have NGN2 that gets turned on by a molecule that makes it so that we're all getting to the same phase at the same time. So it's definitely a caveat to this work that regardless of the cell type we're making, NPCs, neurons, or glia. Well, glia is a little different. The glia, we go into the NPC stage using our protocol, and then we use this media called astrocyte media from Science Cell. So it's not like turning on docs or anything. So there's a little bit variation there that could come into play. But for our neurons and our NPCs, it's all docs driven. And so, yeah, all that variation is going to be eliminated leading up to the experiment.
Thank you.
Yeah.
I have kind of a question kind of in that same early space to
Yeah.
open up other avenues in our thought is that you mentioned specifically, I think, early on that if you try to put stuff together while you're inducing, it's a mess. Um, I think Sally just had to drop off, but
Sure.
her work with Taylor Bertucci, they actually treat individual cell lines with differing levels of dual SMAD to get to a uniform point, which I think is reflecting the exact variation that you're trying to dodge
Yeah.
by doing the induction individually, and then pooling that common neural progenitor state. I guess, yeah, just that's more of a comment than a question, but
Yeah. Yeah.
if feel free to talk about it. I think this gets you to that great advantage of being able to freeze pools and just thaw 50 lines in a single tube, which, practically speaking, is just insanely efficient compared to what
Yes.
if you would have to repool every time you started a new experiment. Um,
Exactly. Yeah.
yeah.
Yeah. So it really comes down to a lot of people are trying to contend with this issue of how do I have balanced pools, right? Um, if I have 50 donors, how do I know there's at least going to be 20 of them a week later? You meaning like 30 don't just drop out because the other 20 just overgrow, right? And so we do a lot of things to try to minimize that. We play to lower density. We pass this before they're really starting to grow on top of each other. We try to eliminate any sort of sense of, cell competition and like the biological definition of cell competition. We try to really eliminate that. But the problem is, one thing we've noticed is that the growth rate in an array matches their growth rate in a village. So
Which is
we,
huge, right? Yeah.
yeah, it's huge because what it means is we just built a village, a 50 donor village yesterday, and we actually took pictures of each of the cells prior to pooling. We took pictures for 5 days. We can calculate growth rates. So we knew before we made the village, this donor is a major red flag because we can predict that within 4 days, it's going to be 50% of the village. So we had two options. Don't include that donor or
Include less of it.
lower proportion. So we put it at 25% of what it should have been, just so it could still be represented, but it wouldn't dominate by the time that we're harvesting, which is like day 6 for this experiment. Now we could do that because we knew each donor's growth rate going into it. If you start with, say,
Right. You
oh,
usually wouldn't know that, right? Yeah.
yeah, not only that, but like their growth rates can change throughout differentiation. So if we know their NPC growth rate or their stem cell growth rate, and we pool the stem cells and then try to differentiate them together towards a certain fate, their growth rates are no longer necessarily going to match what they were when they are arrayed. So we actually lose that predictive power to identify a donor that's going to be problematic down the line. And so all it takes is one donor to really grow
Mess
super
up your
fast.
dish.
It completely ruin an experiment. And so we've been building tools, and hopefully in the next 6 months, we'll have this sort of imaging-based platform. We're calling it Clonalisa, that we've been using to really monitor our cells before they go into a village or before they go into a frozen mini pool to protect us from certain donors taking over and really screwing everything up down the line. But just one last thing in general, I'd say um, if you are going to do any inductions as a pool, my recommendation is just make sure that induction is highly robust across your donors and make sure that you're not doing that for more than I'd say 5 to 7 days, because in that window it's the Wild West and you're going to have a lot of trouble knowing if you're going to have that balance. Some people were saying like we don't care. We'll put 200 lines in there and
And study
it could be
20 or
100.
study
Yeah.
50. Yeah.
Exactly. Be it 50 or 100, doesn't matter. That's enough for us to do these studies. I think that's also a viable approach if you have the people power to kind of, deal with 200 lines.
Some of us are perversely interested
Right.
in the
In
fact
that
that these
step,
cells
yeah.
grow at different rates.
Yes.
And we want to study that actually.
Exactly.
And for those of you who are interested in that topic, the last of these seminar series, Alex Pollan talked, and we had a really interesting discussion about the same issue there, where it seemed like in their paradigm to differentiate dopamine neurons, they didn't see these big effects.
Mhm.
So these big effects, it seems like I'm open to being corrected here, but it seems like that they're associated with more anterior fates.
Mm.
It's potentially true, I think, that although we call it growth, actually it's not strictly growth. I mean, it's not simply cell numbers. Is that right? It could be changes in state and that we're not monitoring. And because if you think of the cells going from pluripotent state to a different kind of anterior phase, that's much more complicated than making a gastroloid.
Yeah.
Right? In terms of the number of developmental transitions that take place.
Mm-hmm.
Potentially, at least. Anyway, it's interesting. It's worth, I think, listening to Alex on the topic.
Yeah, and also say, if you're working with neurons, the quickest you can get them out of the cell cycle, the better you are. Because once, of course, they're no longer proliferating at all, there's no differences in proliferation rates across the donors. And so you can also have a situation where you have a really nicely balanced pool because they were only different, only proliferating for a couple of days. So that's actually how we plan on making our neuron villages from these frozen NPC mini pools, is we pull them all together in the presence of docs, plate them down within 3 days throughout the cell cycle. And so ideally, those 3 days is not enough time for a lot of variation in growth rates to really drive differences in donor balance. And so we can have a nice balanced pool. So I also wonder if that's at play with Alex's work, if he was looking at dopaminergic neurons, if some just were able to get out of the cell cycle, or that they were able to get out of the cell cycle around the same time.
Yeah, no, that's a good point. The dopamine neurons come from the floor plate, and the floor plate has completely different proliferative kinetics from the rest of the neural tube.
Yeah.
So, yeah,
And of course, the
absolutely.
telecephalon that most of us are studying is enormous in comparison, right?
Yeah.
We could talk about this for a long time. Thank you.
Yeah, thanks so much for the return and the new work. Really appreciate it.
Yeah, hopefully we can come back soon with more data, maybe for my trainees. I'll give them a chance to talk soon.
Sounds good.
Yeah, great. Thank you.
Thank you all.
Bye. Thank you.
Ciao.
Thank you.
Bye, everybody.